Today we will be modeling the propagation of an action potential down the axon of a neuron. A neuron is an electrically charged cell that processes and conducts different chemical reactions throughout the body. First, let's discuss the anatomy of a neuron. You have your cell body, the axon, the dendrites, and last but not least, the oligodendrocytes. Oligodendrocytes are a type of neuroglia that provide support and insulation for the axon. They are what create the myelin sheaths for neurons in the peripheral nervous system. The axon of a neuron is the impulse-conducting portion of the neuron. What action potential is a fast-paced action where the nerve pulse is carried electrically through a continual wave motion. For a neuron at rest, there is a high concentration of sodium ions outside the cell, which are positively charged, and a high concentration of potassium ions inside the cell, which are negatively charged. There are 5 phases during an action potential. The first stage of an action potential is where the neuron is at its resting state. At this state, the neuron is charged at -70 millivolts. The second stage of an action potential is called depolarization. This is where the voltage of the cell becomes more positive and less negative. Depolarization causes the voltage-gated sodium channels to open, allowing for the positive sodium ions to come rushing into the neuron. When the gates initially open, the gates only leak in the sodium ions until it reaches threshold at about -50 millivolts. Depolarization continues until the voltage of the cell becomes about 35 millivolts, and at this point, the voltage-gated sodium channels close and the voltage-gated potassium channels open. This stage is called repolarization, making the cell voltage more negative again and less positive. Voltage-gated potassium channels open and close very slowly, causing too much potassium to enter the cell, making the voltage about -95 millivolts. This is called hyperpolarization. Through diffusion across the membrane, based on the concentration gradient and membrane resistance, the voltage is brought back to the resting potential of -70 millivolts. In the final stage, potassium channels close. This process continues repeatedly all the way down the axon based on different stimuli. In myelinated cells, action potential only occurs in the nodes of Ranvier, or gaps between myelin sheaths. These are areas where there is no myelin to insulate, which produces a faster conduction because the impulse can jump from one node of Ranvier to the next. This conduction is called saltatory conduction. Saltatory conduction is found in the peripheral nervous system, which consists of all the nerves around the brain and spinal cord. Neuroglia are the cells that maintain homeostasis and form myelin, meaning that they have saltatory conduction. Unlike saltatory conduction, however, continuous conduction is a more continual conduction where the nerve is bare of myelin, causing the action potential to move in a slower continual wave. This conduction is found in the central nervous system, containing only the brain and spinal cord. Conduction velocity is the term used to measure the speed of propagation along the axon. There is also a mathematical constant called the length constant, which can figure out the distance that an electrical current will travel along the neuron. And there you have it. That's the propagation of an action potential down the axon of a neuron.